I'm so excited to have the opportunity to present the findings of our research here today at the meeting with regard to single mosquito metatranscriptomic sequencing. Now, this project arose out of, frankly, curiosity, as well as limitations that we were noticing in data that were arising from more conventional bulk mosquito mNGS studies that were being reported in the literature. Next slide, please. Now, these studies entailed the pooling of hundreds to thousands of mosquitoes and sequencing them all together in a single batch. And while this provided a wealth of information about populations of mosquitoes around the world and the microbes that are present in those populations, there are a number of shortcomings of these studies that we felt might be addressed by single mosquito sequencing. First of all, these studies required visual inspection and time and labor-intensive sorting of mosquito species to ensure that you had the right host microbe interactions in your batches. Secondly, the studies were typically viral in focus and not taking full advantage of the power of the metagenomic approaches that they were employing. Thirdly, the batch-based approach to these blinded the analysis to any type of co-infections or microbial interactions that were occurring that could influence the pathogenicity or transmission of virus or other infectious agents of interest. Likewise, this population level unit of analysis also affected critical epidemiologic measures and made them challenging or in some cases impossible to acquire. And next slide, this is pretty important because many people consider that mosquitoes are actually the most dangerous animal on the planet. They're a source of emerging and re-emerging infectious diseases responsible for nearly 1 million deaths worldwide annually. And with the changes in global climate in terms of temperature and meteorologic patterns, we know that in the ensuing decades, the habitat suitability for mosquitoes is likely to expand across the globe. And with that, we can expect a concomitant expansion of mosquito-borne diseases. Next slide. In view of all this, we're really interested in looking at how single mosquito mNGS approaches, if they could provide a potentially higher resolution information about the mosquitoes, as well as more epidemiologically relevant information. And to begin to investigate that, we first had to develop a high throughput approach to extract RNA and sequence individual mosquitoes. We worked pretty hard and iterated on that with our partners in the mosquito vector and control agencies, and we're able to spin up that kind of a pipeline. And then once we had that pipeline in place in the lab, we merged that with the existing ID-seq metagenomic sequence analysis pipeline that we heard about much earlier before the talk started, that was developed here at the Biohub and in conjunction with CZI. And what this allowed us to do was to take all those reads that were coming out of the sequence analysis and automate the sorting of host and non-host sequences, assemble the non-host sequences into longer contigs that we could then use to as tools to search for homologous sequences and taxon assignments of all the sequences that were present in the non-host fraction. Next, this allowed us to ask specific questions about what mosquito hosts are circulating, what microbial reagents they're carrying, and if there are any micro interactions, also, and if we could see what they were feeding on in terms of the overall population. Next slide. Now, the starting point for this study was in our own backyard here in California. We worked, as I mentioned, with local mosquito vector and control agency members across California to collect a diverse set of mosquito species, both blood-fed and unfed variants of those mosquitoes. And when we got all these mosquitoes on board, we rapidly extracted them, ran them through the analysis. And then one of the first questions we wanted to ask, if we go to the next slide, is whether we could infer the mosquito species from the metagenomic sequence. And so to do that, we basically took all the individual mosquito transcriptomes, compared them to each other, and clustered them according to their similarity. And when we finished that, we overlaid the clustergram that I'm showing you here with the visual calls that were made by our collaborators at the time of specimen collection. And what was really gratifying about that, as you can see on this diagram, is we have clear delineation of the different genuses of mosquitoes that were represented in the population. And then if you look at the lower gray boxes at the nodes, the leaf nodes of this tree, you can see that our 10 different individual mosquito species that we were able to collect for this study were readily separated. Well, that was really awesome, but I'd be completely lying if I didn't tell you there were some discordancies. So next, we actually found about 7 samples that were discordant between our the visual calls from our partners and our computational calls. And what was really gratifying to us was when we actually dug down and looked at those discrepancies, we found that they generally occurred among species of mosquitoes with overlapping morphological features that were often mischaracterized by visual inspection analyses. So in the end, we feel like we found that our sequence data was very robust and ultimately used that for the speciation of the mosquitoes in this study. And I think this is a really great starting point for future studies to move forward with that, getting rid of that labor-intensive and time-intensive step that I highlighted earlier. Okay, next slide. Well, after we dealt with the host side, we then moved on to the non-host fraction of the mosquitoes populations. And what I'm showing you here is the diverse set of microbes that we could detect across all the mosquitoes in the non-host fraction. Now, this is a tree map, and in the tree map, we're showing viruses in rainbow colors, bacteria in poopy brown colors on the lower right bottom part of the plot, and eukaryotes in the lower left bottom of the plot in green to like blue-gray colors. And the size of the boxes corresponds to the number of reads that we are able to recover for each of these taxa. Now I'm going to go through each of these taxa and highlight a few interesting observations, starting with viruses. So next slide. What we saw was viruses really made up the dominant microbe in these non-host fractions. 84% of the assembled non-host reads that we identified in our data corresponded to viruses. This represented about a dozen different viral families that were very diverse in the types of genomes they had. And within those viral families, there are over about 70 distinct viral species that we actually captured in terms of full genome sequence recovery. And among those, more than half of them were novel isolates that were not detected in California or anywhere else in the world before this work was done. Now, what I'm showing you here is sort of like an in silico version of bulk mosquito results for our study. And the whole point of doing this study was to understand what's going on at the individual mosquito level. And I'm going to tell you about that in a second, but just touch on 2 really high level results that I thought were really important and interesting. So the first thing that we could do once we moved to single mosquitoes was, next slide, ask about how the viruses were distributed among these mosquitoes. So what you're seeing here is a histogram of the number of samples that had different numbers of viral species detected in them. And what you can see very clearly from this plot is that viral co-infections are common. And the implications of this next are pretty striking in that it suggests that or provides a kind of an answer as to why mosquitoes may be such a source of emerging viral infections, in that they have multiple viruses all kind of in the same milieu coexisting together. It provides a really great opportunity for recombination and reassortment among viruses. So that's one particular observation that we saw. And we did a lot of work following up on this to drill down on how much viruses we were seeing in different mosquitoes, how they partitioned across mosquito species, which ones were co-occurring together. And unfortunately, I don't have a lot of time to tell you about that now, because I want to tell you about another really neat result that we gleaned from these data using single mosquitoes. And that's next slide. Understanding what's present in the mNGS dark matter of our data. So every study where you do metagenomic sequencing, there's this whole pool of reads or contigs in our case that when you blast them or look for sequence similarity to them, you find no sequence similarity to anything. And so with single mosquito analysis, it provided us an opportunity to use a simple sequence independent approach to look at these types of sequences and see if we could pull any sequences out there or infer anything about those sequences. Next, we simply just asked the question of whether or not we could detect contigs in the dark matter that consistently co-occurred with viral contigs. Now, this is especially useful in the case where you have novel segmented viruses that you have a sense of how many segments they have, and you only have a subset of those segments. Let me just pull up next a little diagram to show you, kind of give you an idea of what this looks like. So here's a toy example of 6 different mosquitoes, each of them with a set of contigs that were identified as virus through homology searches, and another set, a whole, as I mentioned, a whole pile of contigs that weren't identified as anything because they have no sequence identity. Now, we can clearly see next that there's one group of mosquitoes that have purple virus and some dark contigs that are present in all the other samples. But if we click next twice. If we look at the other set, there's this other set of mosquitoes that have blue viruses. And in addition to the dark matter that's shared by all the other mosquitoes, they also have this gray contig next in the dark matter that is unique to co-occurring with all the samples that have that blue virus. And so this is an example of how we kind of went through all the samples and looked for starting points for interesting novel sequences in the dark matter that might be leads to new and highly divergent sequences that we wouldn't be able to identify through homology-based approaches. Next slide. And so what we were able to do with this was identify two different types of discoveries. One, we could use this approach for recovery of highly divergent missing segments from multi-segmented viruses. So for example, next, we found a series of viruses that belong to the influenza virus family. These viruses the viruses in this family typically have 8 segments, but the viruses that we found had 6, 5, and even 4 segments. After performing our co-occurrence analysis and looking in the dark matter next, we were able to recover a complete set of 8 segments for all of these viruses. And what was really cool about this was when we found those segments, we were able to take those segments and by homology go to other studies where they had also identified only 6 segments or only 5 segments and look into the dark matter of those studies and actually see that, yes, those segments were also present independently in other studies, sort of validating that these are actually real segments. The second sort of application that we use this for next is actually to discover additional gene segments in viruses that were previously not known to be segmented. So here's an example of a narnavirus that's a single segment virus. We had it in 43 of our samples of mosquitoes. It occurred 43 times. We did the dark matter co-occurrence analysis and next actually came up with in each sample identified another smaller segment that also had no sequence identity to anything that was known before, but had all the features of the genomic structure that's seen in the narnavirus. And again, we were able to validate this independently in other studies where narnavirus has been identified. So there's next, we've done this primarily for looking in the dark matter and only for finding viral sequences, but I'm really optimistic about this approach and the simplicity of it being able to apply to not just viral genome segments, but perhaps viral microbe interactions. And let's move to the next slide. Speaking of viral microbe interactions, I want to move on to looking at the bacteria next. So I'm highlighting here the poopy brown cluster in blocks in the tree map. And this just highlights sort of the proportion of samples that, the proportion of reads in our non-host fraction that were bacterial. And a lot of these were generally found, we could only assign them at very high level taxonomic categories, but there was one very large box, I think you might be able to see it, that corresponded to a genus level assignment of Wolbachia. Next slide. This was a dominant species that we could identify at such a low level. And I wanted to just walk through the preliminary observations related to Wolbachia that we found. First of all, Wolbachia is known to be a microbe that is resident of mosquitoes, and we detected Wolbachia only in mosquitoes that we are known to be susceptible to Wolbachia. So that was kind of a nice sanity check that we actually saw it where we'd expected to see it. And 2 of those examples are shown here. On the left, we have Aedes albopictus, which is known to be susceptible to Wolbachia, as well as Culex pipiens. And what I'm showing you here is individual mosquitoes and sort of the fraction of non-host reads that corresponded to Wolbachia in each of these mosquito species. So first of all, you can see that this occurred at a very high prevalence. Almost all the samples had Wolbachia, but each species of mosquito seemed to have different proportions. And next, when we rolled in the virus profiles that we recovered in these samples, we start to see a glimmer of an interaction between the Wolbachia infection and the types of viruses. If you look at Aedes albopictus, you can see a dramatic difference between the types of viruses in the low Wolbachia load sampled relative to the higher Wolbachia load samples. And in the case of Culex pipiens, there appears to be some sort of mutually antagonistic relationship between abundance of viruses and the amount of Wolbachia present. So, we have very small number of samples here, so it's very difficult to say anything statistically significant. But I think the fact that we can start to see a glimmer of these interactions is really promising for the utility of doing single mosquito sequencing and probing these interactions going forward. Okay, next slide. I will close out the discussion talking about what we saw among the eukaryotes. So again, this is the lower left-hand corner of the tree map that we showed earlier. we actually recovered diverse taxa ranging from plants to known microbes of mosquitoes and ultimately to vertebrate taxa. I'm really just going to focus on the vertebrate taxa because what was really interesting about these, next slide, is that we saw these vertebrate taxa only in the samples that had been collected and were identified as blood-fed on collection. And in fact, 45 out of 60 of the blood-fed mosquitoes actually had detectable vertebrata RNA sequences in them. And so what I'm showing you right here is the level of the lowest common ancestor at which we could assign for these different vertebrata taxa amongst the blood-fed samples across the data in our sample collection. And interestingly, among these, almost 1/3 of the blood meal positives showed some co-occurring detectable microbes. And what was neat about that was, in the birds and the aves, the microbes that we see are known microbes of birds. And likewise, the bacteria and novel virus that we saw in the pecora samples corresponded to known pathogens of these types of animals. And next, I think just what this underscores is that single mosquito sequencing can also elucidate candidate reservoirs of both known and novel microbes. So I'll finish it out in the last slide next by just summarizing. I hope that I've convinced you that single mosquito metatranscriptome sequencing provides a whole raft of unique and actionable insights that can be used to really help us control and follow better both mosquitoes in circulating environment as well as the microbes they carry. And next, I think, in thinking about the impossible and technology, we're seeing sequencing costs continue to decrease, and our ability to automate and miniaturize these reactions, I think, makes a number of potential applications really a potential reality in the next 5 to 10 years. In particular, I see rapid early stage investigations of emerging infectious diseases as an important area where we could contribute with this type of analysis. And likewise, I think this sort of work could help guide or augment conventional seasonal surveillance programs to make sure they're on tap with what's really circulating in the environment. And lastly, I think this deeper analysis of this work could help us to identify additional candidate biological control agents like Wolbachia that are being used in the world right now to control the spread of mosquito and vector-borne diseases. So with that, I'll acknowledge in the next slide my collaborators on the project. There's a host of biologists and data scientists from the Biohub and CZI that participated in developing this program. Of course, I had tremendous support from leadership, and we couldn't have done any of this work without our intrepid collaborators at the mosquito vector and control districts in California who participated in this study. And I'll just close with a thank you, next slide, and take any questions if there's time.
Were you able to detect any instances of recombination among viruses that you detected in the sequencing, given the small sample size?
We weren't able to detect that in this particular data set, but it would be something that would be really interesting to look at in a much larger sampling set. It's a great question.
And then we have a question of how well will this scale for pre-processing mosquito samples for mNGS? So, is this something that could conceivably be put into a package that could be used by county departments of public health, for instance?
I don't think we're there yet. And I think if you would ask me where the next step that needs to happen to help with the feasibility of this is actually the mosquito collection phase. If there were a way we could collect mosquitoes into some sort of plates, that would be great. I know we have some CZ Biohub investigators who've been doing some amazing work, Manu Prakash, looking specifically at collecting more saliva of mosquitoes. But there's a whole lot of smart mosquito traps out there that could be good candidates for us to partner with to really bring this from the field into, say, a public health lab or a reference lab where we could really do this in high throughput.
And I'll sneak in one yes/no question. Can this be readily extended to ticks?
Yes.
Or other vectors?
Yes.
Terrific.
Doing that.